Bioinformatics Micromodules, Short Read Alignment. In this module, we introduce the short read alignment problem. In the previous modules, we assume that there are some sequences provided to us, based on which we can perform different types of analysis, such as sequence alignment and phylogenetic tree reconstruction. How are these sequences actually obtained in the first place? In the case of DNA sequences, they are commonly obtained by a process called sequencing. Suppose originally we have a biological sample that contains some DNA sequences. Each sequence may have multiple copies in the sample, say from different cells of a tissue. In the illustration, the DNA sequences are shown in gray to emphasize that their actual contents are not known to us beforehand. During a sequencing experiment, the DNA sequences are first cut into short fragments, and then the fragment ends are sequenced to find out their actual order of nucleotides. In high-throughput sequencing, many fragment ends are sequenced simultaneously in parallel, and therefore, at the end of the experiment, many such fragment end sequences are obtained at the same time, each in the form of a text string called a short read. Current high-throughput sequencing methods, also referred to as next-generation sequencing methods, usually produce reads not longer than 250 base pairs. There are many nice videos that explain how high-throughput sequencing is performed, and you are welcome to learn the details, but such knowledge is not required for this micromodule. What we focus here is that, given a short read, we want to find out the location of it in the original sequence before fragmentation. Usually, we do that by looking for the sequence of the short read from a reference sequence, such as the human reference genome. Based on this description, you can see that the short read alignment problem is basically a pairwise local sequence alignment problem. If you have already forgotten some concepts related to the sequence alignment, this is a good opportunity to refresh our memory. Pairwise here means that we have two input sequences, namely a reference sequence and a short read. The alignment is a local alignment because only a small part of the reference can be aligned to the short read. Imagine we have performed DNA sequencing from a human cell sample. Then each time, we are aligning a short read, which is up to hundreds of base pairs long, with the reference genome, which is billions of base pairs long. We are not doing this alignment only once in total, but rather once per short read produced. Since current sequencing machines can produce millions or even billions of reads in a single run, we need to perform the alignment millions or billions of times. Let us see a simple toy example. Suppose we have a reference DNA sequence S, which is TATACATTAG. Once again, in reality, S can contain billions of nucleotides. Suppose further that we have produced some short reads. Here we assume that 7 reads have been produced, each having 2 to 3 nucleotides. In reality, there are millions or more reads, each with up to hundreds of nucleotides. Now we align the reads with the reference to find out where they came from. In some cases, such as ACA, exactly one perfect match is found, and therefore the read likely came from a DNA fragment that started there, especially when the read actually contains hundreds of nucleotides and it is difficult to get a perfect match by chance. On the other hand, the read AT has 2 perfect matches with the reference. This non-unique alignment is due to a combination of short read length and repeating content in the reference. We should also notice that the read TTC does not have a perfect match with the reference, which can be due to sequencing errors or differences between the reference sequence and the actual DNA sequence. Finding out these differences is in fact precisely what we want to achieve in sequencing. However, if we have used a good reference sequence, we can expect that most of the reads have a perfect or near perfect match with the reference. And this is important for the methods that we are going to discuss in these several micromodules. Finally, please also notice that some reads can be identical and some positions of the reference may not be covered by any of the reads. In the coming modules, we're going to use this reference sequence S again and again, and therefore it would be great if there is an easy way to remember it. The way that I recommend is to pronounce it as tet a cat tag. Hope that next time when you remember these beautiful cat tags, you'll recall this reference sequence, TATACATTAG. By now, you may be already wondering why we need to study short read alignment as a new problem. Isn't it again just sequence alignment? For instance, why can't we use dynamic programming? The simple reason is that dynamic programming is too slow. If we want to align L short reads, each of length m, with a reference of length n, the time complexity is order LMN. Remember that both L and N are very large numbers, and therefore practically it would take a very long time to do all the alignments. Then what about using BLAST or other heuristic alignment methods? Although heuristic methods are fast, they may still be not fast enough. This is because these methods allow different types of mismatches and indels, but such flexibility is not needed for most of the short reads, which have a perfect or near perfect match with the reference. By making use of this important property, we hope to achieve optimal alignment efficiently for most of the reads. The key approach to achieving efficient short read alignment is building an index of the reference. The term index means a data structure that facilitates efficient data lookup. For example, in a book, the index is usually in the form of a list of keywords and the corresponding page numbers of the pages on which the keywords are mentioned. Here, we want to have an index such that given a short read, the locations of it in the reference can be efficiently retrieved. We can easily imagine that there are requirements for a good index. First, it should not take up too much space. Second, data lookup should be very efficient. Third, the time needed to construct the index should also be reasonably short. But actually, long index building time is more tolerable than long data lookup time because the index is usually built only once and reused many times. Finally, if the data being indexed can be updated, the index should also be updated accordingly in an easy way. However, in these several micromodules, we will assume that the reference sequence is fixed, and therefore we will ignore this updating requirement. In the coming few micromodules, we will introduce several indexes for short read alignment, including suffix trie, suffix tree, and suffix array, and the FM index that is based on Burroughs-Wheeler transform.